Welcome to Recovering Evangelicals, a podcast for people who were once very comfortable in the Christian faith until the 21st century intruded and made it very hard to keep on believing. I'm your host, Luke Jansen.
And I'm Scott Dyer.
Last week we started this mini-series on intelligent design. We introduced the idea itself and our own personal positions on it. Next week we're going to talk to one of the intellectual leaders and a figurehead in the intelligent design movement, Dr. Michael Behe. But before we hear him defend ID, we need to make sure listeners understand some of the basic terminology and basic scientific mechanisms. Having watched and heard many ID advocates make their case, I've learned that one thing they exploit very well is their ability to talk far above the understanding of their audience, a general audience. And they show well-produced, glitzy images and videos to invoke a sense of awe and wonder. But also, I think, to get the audience to drop their defenses. And I'm not trying to accuse them of malevolence. I'm just saying these videos, I think maybe it's the TV age that we grew up in. Once you sit in front of the TV, you just kind of sit back and your brain turns off and stuff goes in. And I think that's what's going on. What do you think, Scott?
Well, yeah, knowledge is a spectator sport these days, in a lot of ways. Yeah.
Okay, so as I said, the intent, or at least the result, is that they get their audience. The audience is left having to trust what they're saying is true and accurate. So I want to try to demystify the genetics and cell biology for our audience. I'll just give you some tools that you can use when you hear them speak. I want to give you some of the background that you'll need to properly evaluate what ID proponents are saying. And Scott, I want you to be the stand-in for the audience. As I walk through these concepts, Scott, I want you to be the audience and ask any non-expert kind of question that comes to mind. You up for that?
Yep, no problem.
Okay, and to the listener, to the audience, I want you to channel Scott. I want you to see the concepts I'm teaching through his eyes and his questions. He's going to interrupt at any point that he says, okay, Luke, this is not quite making sense. And realize that this is going to be a reasonably academic talk aimed at a non-expert level. This is not going to be a theological episode. So if you're wanting a theology-heavy episode, you might be a bit disappointed in this episode, and you might decide to quit halfway, which is your prerogative. But I think you'll find next week's episode a bit easier to follow if you do hang in there. But it's your decision.
Yeah.
Okay. Again, next week we'll be hearing from Dr. Michael Behe, a globally recognized proponent of ID. And for most people who know anything about him, as soon as you hear his name, a few words come immediately to mind. Words like bacterial flagellum and irreducible complexity and maybe mousetraps. And in both cases, he'll be talking about parts of machinery, the physical things that make up the flagellum, which moves the bacterium around, or the mousetrap, which is his analogy. So let's talk about how cells make those parts, those physical things, and how it's the physical structure of those parts that gives them their functionality.
Yeah, certainly Michael Behe and some of the things you've mentioned have become very popular ideas. And any Christian who has any interest in the evolution debate will definitely point to those things. So it's important to dive into those.
Okay. Now, almost everybody knows that the story begins with the genetic code, that the long stretches of the G's and C's and A's and T's. We've all heard that, and most of them will know that the string of those four letters somehow translates into a long string of amino acids, like beads on a string, that in turn make up proteins. I think that much most people have heard. That's not news to most people, right, Scott?
I don't think so. I think, especially with things like, the DNA analysis, 23andMe. And Ancestry.com is another one. Yeah, well, you can buy these kits and send in for your DNA and find out that guess what, you're really half this or half that, or
right.
Yeah. So I think there's a very strong awareness. It's become part of our parlance.
Right. Okay. So I don't want to get into that decoding process right now. Many people have already heard much of the basics of genetics, and maybe we'll do it another time, but not now. It's not crucial for what we want to do right now, which is, again, talking about how proteins form three-dimensional structures, because it's that structure that gives them their function. The genetic code produces these long strings of amino acids, which, again, proteins, and somehow those strings of amino acids become the parts of cellular machinery. A little nanomachine moves along the strand of DNA and squirts out a stream of protein. I want you to visualize this, a long strand of DNA, and a little bot sitting on it, moving along, and out comes a stream of proteins, a series of amino acids strung together like beads on a string. And so, Scott, I want you to imagine you're holding a tube of toothpaste under the water. You're at your kitchen sink or a bathtub full of water, and you gently squeeze the tube, and you just keep gently squeezing the tube for a minute. What happens?
Well, the toothpaste would just kind of go all over it seemed to me you'd kind of see a string of toothpaste, slithering through the water.
That's the idea. Exactly the idea. You see this long string, the strand of toothpaste. In this case, maybe a better example I should have used that occurred to me was people use this at parties. It's a can, an aerosol can. You squeeze it out, and out comes this silly string.
Yes.
And so you can, same thing. You just push the button down, and out comes this long strand of, I don't know what it's made of, but silly string. I think we've all seen that, at least on TV. Now, that string, or the toothpaste, it begins to fold over on itself. It just becomes a glob, and it forms loops and folds that just pile on top of each other. All right? It makes this amorphous blob, looks like spaghetti in a pan. The only difference is that this spaghetti is one long single strand that just has all kinds of loops and folds piled up on top of itself. So can you visualize that big strand of spaghetti or the silly string in a pot, Scott?
Yeah, for sure. Especially the silly string.
Okay. But now we come to a very important concept. Imagine we don't just form an amorphous blob. But maybe we can begin to force the string of toothpaste or the silly string to fold in certain predictable ways. And in so doing, they make predictable shapes, which again is where they get their function. So that we can make useful things like hammers and nails and beams and rafters, cutting tools, a long list of structural components and functional machines. So let's start with something that everyone's familiar with, how we're going to shape this blob of protein, the silly string in the pot. We're going to use a thing called electrostatic attraction. You've heard the phrase opposites attract, right, Scott?
Yep.
Okay. It's a fact of physics that positive charges want to get together with negative charges. They're opposite. There's a strong attractive force between those two. So this is what's going on when you pull the socks out of the dryer. They're stuck to the towel. You pull out the towel, and there's the socks sticking to the towel. That's exactly what's going on. It's electrostatic attraction. By tumbling around in the dryer, charged particles have been pulled off of the socks and accumulated on the clothes, and so they cause the sock to defy gravity. The sock sticks to your shirt or your towel so that you actually have to peel it off. You can also do this with latex balloons. You can rub the balloon, an inflated balloon, on your shirt, and then you can stick it to a wall or to a person. Have you done that, Scott?
No, for sure. and I've also rubbed my feet on the floor and then shocked myself out of doorknob or something.
Well, that's half of the point I'm trying to make. That's the separation of charges, and then you get the sudden shock when they come back together again. But you can separate those charges and hold them separate so that the one side is positive, while the other side is negative, and that's what's holding the sock to the towel, for example. And that's the point I want to get at, because we can do that with our silly string or our tube of toothpaste, the spaghetti. The same thing can happen at the molecular level. The cells, when they're putting out that long string of amino acids, the toothpaste out of the tube, they can incorporate certain amino acids into the long chain of the developing protein. And some of those amino acids that they're putting in there at the right places, they have a positive charge. It's just part of their nature. They're always happy. They're a sunny amino acid. They've got a positive attitude. And then other ones have a negative charge. It's just part of their chemistry, their physics. They're just having a negativity in contrast to the positive ones. So the cells will incorporate a series of positively charged acids here and close to a series of negatively charged amino acids over there. And then the emerging protein actually folds back on itself so that the positives and negatives can join up and hold it together. That's one of the ways that you can tell this string of spaghetti or the toothpaste to fold and connect at this point by having positives here and negatives there.
Right. Okay. Makes total sense.
All straightforward, right?
Yep.
Okay. So that's what happens when you insert opposite charges in the right places, a bunch of positives here and negatives there, and they produce an attractive force. But the opposite is also true. You can insert a whole series of positives here and positives there, and that's going to push those two parts of the protein apart. The toothpaste coming out of the tube is not going to stick there. If it's got positives in this one part and in the other part, they're going to actually be pushed apart. You can do the same thing with negative charges. Have a whole string of amino acids in one part and on the other part. those two parts are not going to cross and join up together. They're going to be pushed apart. This is exactly what you see, for example, when people they get static hair or flyaway hair, or they go to a science museum and they put their hand on a Van de Graaff generator. You must have seen those pictures where people are standing on this thing. They got their hand on a big metal ball and their hair is sticking up on its end.
Yeah.
So the hair is a strand of protein. It's just a filament of protein, and those strands develop a negative charge on them, or maybe it's a positive. I'm not sure which way the polarity goes, but they all have this same heavy, let's call it positive charges. All the hairs get positive charges, so they just all begin to push away from each other, and that just forces them to all stand up on end as far apart as they can possibly get. And that's what gives you that frizzled look. So using these charges, you can develop very strong attractive forces that hold sections together like a rivet, as well as forces that push the protein parts apart. The different loops and folds that develop, they get pushed apart. They do not come together or stay together. Another analogy I think is helpful is like two magnets. When you line up the north pole of one magnet and put it up against the south pole of the other magnet, they suddenly clank together. You might even pinch your finger in the process, and you have to work hard to separate those two. But if you now do the opposite, you put the north pole of one magnet against the north pole of another magnet, you have to work really hard to get those two to connect.
Yep.
It's the same principle. So by incorporating amino acids in the right places that have positive or negative charges, you can then introduce attractive, and we call them repulsive forces. They push things apart. That begins to shape the molecule. That's one of the first things that happens. But there are many other forces, and unless you have questions, Scott, I want to move on to another one.
No, that's great. Yeah.
Okay. All right. So one kind of force doesn't involve attractions or repulsions between positive and negative charges. Again, we call those electrostatic forces, but instead how they interact with water. We call these hydrophobic and hydrophilic forces. And so a crude analogy that you can use to illustrate this is a piece of paper stuck to a window. You just make it wet and it sticks to a window. And that's a bit of a combination of what I'm referring to here, this hydrophilic attraction, as well as some surface tension. But that paper wouldn't stick to the window, or at least not well, if the window was all greased. So the paper is wet and hydrophilic, and you're trying to put it on a window that's all greased, that would be hydrophobic. It just won't stick as well. But a greasy paper would stick to the greasy window and not to the wet window. So basically I'm trying to show that in the same way you can have positive and negatives interacting, you can have water-loving and water-hating molecules or parts of the molecule. Yes. And some will stick together just like the positives sticking to negatives. Some will push themselves apart. Yes. Same idea.
Which is, by the way, why if you, I used to be a marine aquarium enthusiast, even though I don't have one right now. But one thing we do in marine aquariums is we call it protein skimming, where we basically, we fire bubbles up through a tube, and the surface of those bubbles will attract the hydrophobic end of a protein, and it'll attach to the bubble, foam up to the top, and that foam becomes kind of a scum with all that protein in it. And then we just kind of skim that off. It's called protein skimming. And so we remove protein out of the water, which if it builds up could cause, a lot of problems in our tanks. So it's a very interesting process.
Right. So you've got this electrostatic attractions and repulsions, and you've got this water-related attraction and repulsion, the hydrophobic and hydrophilic interactions. Another strategy that the cells will use to mold the proteins into certain shapes is essentially to put a nail into the long string of toothpaste coming out of the tube. It'll, as where you get a certain fold, it drives a nail in there between two parts of the emerging, well, in this case, protein. You actually create a physical connection, a physical attachment, and it's kind of like stapling two pieces of paper together using staples. You're actually physically, stapling them, connecting them. And there's all kinds of examples of this. One is often referred to as hydrogen bond sharing, hydrogen bonding, where one amino acid in one part of the protein, it grabs onto the hydrogen from somebody else, and they start fighting. It's mine, it's mine. They're fighting between the two of them. So they're connected by grabbing onto this piece of hydrogen, and it's kind of like a tug of war. It's a fairly loose but strong enough of a force that it actually can hold certain parts of that long, messy strand of toothpaste, hold it together in that one spot. There's another way that the cells can do this, which is much stronger. We call this disulfide bond formation, basically sulfurs. One amino acid can have a sulfur, another amino acid can have a sulfur, and it's just through the chemistry of this working together, the two sulfurs will actually physically bond. They form a very tight and hard bond. So you're, again, putting a staple between the two strands of the protein that have these sulfurs. You connect the sulfurs, you connect the strand of protein.
So is this how proteins are bonding together?
Yes. One part of the protein, again, a protein is a long stretch of amino acids. Right. One of the amino acids has a sulfur, and another amino acid in a distant part of the protein, which just happens to be in the vicinity. As the protein folds and the loops form and it falls on top of the other loop, and you got these two sulfurs, they make a connection, they bond. And now you've got a rivet that holds those two distant parts of the protein together.
Okay.
And so if there was some looping involved in that, now you've stapled the loop into place. So just to belabor the point then, so the hydrogen bond formation is like two people fighting over something they're holding in their hands, while the disulfide bond formation is like putting a set of handcuffs on the two of them. One is a loose connection, but still strong, somewhat, and the other one is you can't break that. Well, under the right conditions you could, but... So for example, again, another fun fact, those of you who you have your hair made curly or not, you do that using chemicals that break, well, in part, by breaking the sulfide bond formations. Your hair can be curly because there's a whole lot of sulfur bonds that are bonded to other sulfur-containing amino acids, and you have to use what they call a reducing agent to break those sulfur bonds, and now you got straight hair. Or you can use an oxidizing agent to give you curls. So
my wife will really appreciate this podcast because she has curly hair and she straightens it every day.
Okay. So that's two ways that I gave you there of how the cell can introduce amino acids in the right place, in the right way, that they actually make a physical connection that then holds the protein in that shape. Once the loops form by electrostatic interactions and hydrophilic and hydrophobic ones, you can kind of staple it in place using these hydrogen bonds, using these disulfide bridges. There's another one that we can talk about. This is on a much larger scale, though, when a protein forms. When you take a few steps away from the protein and look at it, all those little loops and folds can form little knots. Together they form a little knot or a ball in the string. And they can fit into a hole that was created in another part of the protein. That you can have other electrostatic forces and hydrophilic, hydrophobic ones that cause the protein in a different part to form essentially a big pocket, a hole. One is a little ball, the other one's a little hole, and you can fit the ball through the hole. It's like putting a button through a shirt. It's exactly what I'm talking about. You got a little button that you just push together through the hole in your shirt, and now you've held the shirt together in that shape. The snaps on a jacket, the heads and grooves of puzzle pieces. It's the same idea. One fits into the other and then holds the piece in place. Scott?
Wow. So, it's just fascinating to me listening to you. So all these different types of bonds when these proteins are bonded together in these various ways, are they in essence now building a new protein or
well, I guess technically no, because protein is just a long stretch of amino acids. So if it's the same stretch of amino acids, the same sequence, now it's just folded over. It's still a protein, but it's not a folded or a properly folded protein.
Okay.
So for the listener then, if you caught up, we've just talked about a whole range of forces that can be generated by simply the amino acids themselves, their positions in this strand of protein, which I'm equating to the toothpaste that came out of the tube or the silly string that came out of the can. You get this long string of whatever it is, and in the case of the proteins, certain amino acids here or there or the next place can set the stage for hydrophilic, hydrophobic, electrostatic interactions, hydrogen bond formations, the sulfur bonds, and even this ball and socket, puzzle piece, the head and the groove of the puzzle pieces, the button through the shirt. All of these can create forces that pull this ball, this whole string of loops and folds into the form of a certain form that you want to predictably make. And what you're going to get then is these gigantic globs of protein with odd and diverse shapes and sizes, kind of like pieces of popcorn. If you see a bag of popcorn, throw it on the ground, and you can see all these pieces of popcorn, all with different shapes, and you can visualize those as individual proteins that are held in that shape simply because of where those different amino acids were in the protein. And those amino acids are generating these forces that we talked about. And it needs to be said, in case it's not obvious, that the position of those amino acids in the protein was determined by the gene, right? The structured DNA. The DNA said put an alanine here and put a cysteine there and put it says where all the different amino acids go. And once that string comes out, then all these folds and loops start to pull together and rivets begin to be driven in, and you can form different shapes. We're going to talk a bit more about that. Any questions, Scott?
No, that's great. Yeah.
So we've talked about these forces in the proteins that were created by the amino acids in those proteins. And the forces that we were talking about are basically how amino acids in distant parts interact to hold the protein into a certain shape. That's what we've talked about.
Right.
I now want to take a look a little bit closer, and now we're going to look at each amino acid in context of the guy on either side, the amino acid to the left and to the right of any given amino acid. We can talk about how whether the one on the left or right is this amino acid, that alone will determine, in part, the shape of the protein. Let's talk about how that happens, okay? And to do this, I want you to imagine each amino acid like a piece of Lego. You know, those building blocks that people buy for their kids. I think everyone knows what Lego pieces are. You think, Scott?
Oh, yeah.
Yeah, okay.
I think so.
Okay, and we'll start with only the long and the straight lengths of Lego. We all know that there's many different shapes and sizes of Lego. Let's start with only the ones that are relatively long and they're straight. Not the short pieces, not the ones that have a bend in them, a kink in them. And just for the moment, let's just stop thinking about electrostatic, hydrophobic, hydrophilic, all that kind of stuff that we talked about, because those are actions between distant parts of the protein. I want to talk about amino acid interacting with the guy to the left and the guy to the right. When you start stringing them together, what shape they'll form. So, Scott, you've got in your hand a whole handful of these relatively long and straight Lego pieces, and you start connecting them end to end. What shape will they create?
You'd think a straight shape, like
a straight line, like a pole or a filament.
Right.
So now I want you to imagine doing the same thing with a Lego piece where each piece, all the ones that are in your hand, have a 90-degree bend in them. And you start connecting those pieces together. What are you now going to form?
Right, a square, maybe.
What if those pieces didn't have 90-degree bends in them, but they only had 60-degree bends in them? Can you think what would happen then?
60 degree. Well, then you get rectangle? No? I'm not good on geometry.
Well, okay. So it turns out you put together 4 pieces with a 90-degree bend, or you'd put together 6 with a 60-degree bend. And a shape with 6 sides is a hexagon.
Hexagon, okay.
Right?
Okay.
Yeah. And then if you did that, instead of them having a 60-degree bend, let's change it again. Let's make it a 45. Half of the 90. Right. If you started connecting those together, can you visualize what you'd have at that point?
Octagon? No.
An octagon with how many pieces put together?
8.
8 of those blocks will give you an octagon. Just simply, you have a slightly different shape right there because simply of that one bend connecting each individual piece, you now created 3 different shapes, a square versus hexagon or an octagon.
Right.
Now, this is where it gets really fun, and it shouldn't be too much harder. We've been building only in 2 dimensions here. We've been building flat on the table and connecting them flat on the table. But let's now start talking about a 3rd dimension, building in the vertical. I want you to imagine that you don't have to create those shapes flat on the table, but there's enough flexibility in the blocks and in their connections that you can start to lift them slightly off the table as you connect one to the next. And so let's go back to our example I started you with, the 4 blocks with the 90-degree angles that you connected into a flat square. But now before you connect the 4th piece to the first piece to close the square, what happens if you gently lifted the 4th piece by putting, kind of an upward bend in the whole structure? And instead of connecting that 4th piece to the first, you now connected it to a 5th. And then you added a 6th and a 7th and an 8th. What do you start to see happening in your mind there?
An Escher drawing.
Okay, I can see why you'd say that.
Kind of like a stairway,
Essentially, yes, you would have a stairway.
A spiral, like maybe.
Well, and it's close to a spiral as well. Basically, what you've essentially done is you've stacked one square on top of the underlying square.
Right.
And now, if you do the same thing, go another round. You continue with that slight bend in there, and you add another 4. Now you got 3 squares on top of each other, and 4, and then 5, and 6, and you end up stacking a whole series of squares on top of each other. how would you describe that shape?
A tower?
Sure, perfect. Yeah,
Yeah.
it'll be a
Okay.
square pipe essentially,
Yeah.
right?
Yeah. Yeah.
It would
Yeah.
look like a tower, but it would have a hollow in the middle of it.
Right. Right.
And if you did the exact same thing with the 6 pieces that had the
Right.
6 degrees, again you'd have a tower, but this one now
Right.
would have more of a hexagon shape
Right.
to this
Right.
tower. And if you did the 8 pieces at 45
Yeah.
degree angles, you build this tower, you get basically a larger, fatter pipe with 8 sides on it.
Right. Right.
Okay. So now you can see how it might be possible to squeeze the toothpaste out of the tube in such a way that you build tubes or pipes instead of a filament. We talked about this long filament, this long
Right.
string that kind of collapsed on itself, and he had loops and folds. But now you squeeze it out in a way that now builds these towers.
Right.
A self-reinforced filament, a beam, a strong beam. And where this becomes really useful, if you think about it, it's kind of like building, now I'm going to jump to a different metaphor, copper tubing and copper wire. If you tried to make something with copper wire or a copper tube, you'd have different strengths in the structure that you're making. Try to imagine building a chair out of copper wire or a
Right.
chair out of copper tubing. You could probably sit in the chair that you made with copper tubing. But copper
Yeah.
wire, it's going to collapse, right?
Yeah. I was just going to say, it's kind of when you start doing that, you're creating a structure,
Exactly.
and that structure is going to have structural properties, right? It's going to be, might be flexible, it might be rigid, it might, and so on and so forth.
Right. So basically that is what we call, you used the word actually in the beginning, I think you did, you talked about a helix, right?
Yes.
You're building a helix structure, and this is one of the ways that you might hear biochemists talking about an alpha helix. You're basically squeezing out this toothpaste in such a way that it forms this, you use the word tower, it forms this tower that's self-reinforced, and this is where you can now again bring in the hydrophilic and hydrophobic interactions
Right.
that are going to hold that tower, all those pieces together. And the actual spiral of the tower is determined by the one amino acid interacting with the guy on the left and the guy on the right. And holding the whole structure together would be amino acid number 18 holding onto amino acid number 25 and forming a hydrogen bond or an electrostatic bond. So basically now you can see how these forces can work together with amino acids on either side of each other and with amino acids in completely different parts of the protein to give you these very amazing structures, right? We talked about filaments and poles and tubes and chairs and things.
Right.
Okay, so we've talked enough about this helical structure, the tube, the towers that you talked about. Let's now talk about a different structure. I want you to imagine again, Scott, that you've got a handful of straight Lego pieces end to end. Any number, let's just say 11. You've got 11 of these together and you string them together end to end. What do you get?
A straight line, like
A straight line of filament, exactly what you said before.
Yeah, filament, yeah.
So now, at the end of that, we're going to add a 12th piece. This piece has not a 45 degree, not a 60, not a 90, but a 180 degree bend. All right, so you've got these 11 together in a long straight line, and you stick a 12th one on that has a 180 degree bend, and now you add another 11. What are you going to get?
You get a rectangle, I guess.
Essentially, yeah. You'll have a very narrow rectangle. It's going to be
very narrow rectangle.
Two filaments, one immediately beside the other one.
Right.
Okay, and then again, you could reinforce that. These two filaments side by side, reinforce them with hydrostatic or hydrophilic or electrostatic interactions. But then you add another, to the end of that, another 180 degree bend and 11 more straight pieces.
Right.
And then another 180 degree and 11, and another and another. So instead of two filaments side by side, you'll have 3 and then 4 and 5 and 6 and 7 filaments side by side. What shape are you seeing there?
Just you basically have a bunch of filaments side by side, kind of a mat, like
a mat. Sure. I was going to say a sheet of paper, a plank. These are all things and same thing that you said, a mat. That's what you would start to see.
Right.
Just simply by having these amino acids, 11 that are straight and the 12th that's always 180 degrees turn, and just repeat that over and over again, back and forth, you're going to get this sheet. And so biochemists might use a term that I've heard many times, and this is really simplifying things overly, but they'll talk about beta-pleated sheets is another common structure that these amino acids form simply because you string a certain sequence of amino acids together, they form these beta-pleated sheets. And then those sheets can be reinforced with, again, hydrostatic charges and hydrogen bonding, and it's really amazing.
Yeah.
So if you've kept up, and I'm speaking to the listener now because I know Scott's kept up, just simply by the whether you place this amino acid here or there and what's next to it, we've talked about how you're going to build up poles, the filaments. Yep. We've talked about pipes and beams and boards and sheets. You start to see how the sequence determines the shape that you're going to get.
Right.
So let's not try to build on that. Let's try to make something really cool. We're going to make a screw, okay? Or a bolt, a screw or a bolt. Again, simply from a long bead on a string, a toothpaste from the tube, silly string from a can. And we're going to use all these forces and connections that we talked about. So let's start by making a helix, a tube or a pipe, by stringing together amino acids that have these 90 or 60 degree bends in it. And you keep on doing this in the vertical direction. You build that tower that you talked about, Scott. And we'll keep going till, let's just say, for argument's sake, you're 3/4 inch high. You've just continued to add these amino acids in that helical pattern, the 90 degree or 60 degree bends at each point. And now we're 3/4 high. We've got this cylinder that's 3/4 of an inch high. Then we reverse direction. We still stay with the same strand that we built up into the form of a cylinder, but now we start to change using different amino acids, a different sequence, a slightly larger diameter spiral all the way down on the outside of that cylinder. So we built up a cylinder 3/4 of an inch high, and now we're going to start working our way down with a slightly larger diameter and maybe a bit more distantly spaced out lines. Let's use that word. So now you can see the beginning of the threads that form around this central post. And you go all the way down to the bottom, and you're still squeezing out the toothpaste. And once you get to the bottom, then you give this big, massive squeeze to put a glob on the end of it. And now you've got a screw or a bolt. From simply that one long strand of toothpaste or silly string. And to get back to our proteins, just by simply stringing together certain amino acids in a certain way, they will form this post and this helical thread, and then this big glob in the end. That's how they're going to form that. And now you've got a screw or a bolt.
Really? Yeah.
So it's really amazing how those simple ideas can give you such complicated shapes.
Yeah, I think of the analogy of one of the most popular, computer games. Not really a game in a way, but Minecraft. And that's the basic concept behind it. You have a few very basic building blocks, and then what people build out of it is just mind-boggling. What they can build, and yet those building blocks are far more crude than the building blocks we're talking about. They can build very complex structures.
We're just talking about toothpaste or silly string, right?
Yeah,
it's just simply a long strand of something, and in the case of what we really want to be talking about is protein. It's just a string of amino acids. That's all it is. But they're carefully selected. Boy, that's a design-loaded word, eh? Yeah. So we'll have to unpack that with Michael and Jonathan. So we've just finished talking about a screw. I want to now talk about something a little bit more closer to the subject at hand here. We're talking about cells. Back in the day, when I was doing research in the university, one of my areas of expertise was ion channels, which are really just simply holes in the membrane that let charges in and out, ions. You might have a potassium channel that just moves potassium ions in and out. Same thing with the sodium or calcium channel. It's that movement of those ions, those charges across membrane, that gives you the electrical signals that your cells work with. So it's really just go ahead.
Remind me what an ion is. Just remind me what.
Well, an ion is a charged molecule.
Okay, has a charge on it. Okay.
Yeah.
Okay.
So one common structure for ion channels is basically 4 protein globs holding together. All right. So I want you to imagine 4 people standing, holding hands to form a really tight circle. They've kind of pulled in their hands, and they're basically standing chest to chest, all 4 of them. And so they form this tightly knit 4-sided structure. And if you were to drop a box of balloons on their head, those balloons would just bounce off their heads. They wouldn't go into this structure, which again is a closed structure in the membrane of the cell. But now I want you to imagine that these 4 people just suddenly now stretch out their arms while they're still holding hands. They stretch them out, So they have to take a step back, and they're still all holding hands, but now you've got this four-sided structure, big open structure, that if you now drop the box of balloons on their heads, all those balloons will start dropping down past their head, past their chest, and down to the ground to their feet. So that's the channel now open, simply by literally just taking a step back. Each glob takes a step back and it forms together a hole in the membrane. That's all an ion channel is. All right. And then it has, we don't have time to get into it, but that four glob shaped structure, which is an ion channel, has ways to tell whether the balloons that are falling down through the center, whether they're potassium ions or sodium ions. We just don't have the time to explain what that's all about.
Right.
But,
Okay.
But I just want you to imagine how even this structure, which is really complicated, it's a potassium channel that moves potassium ions through the membrane.
Yeah.
All it is basically four globs holding hands together, and they take one step back and open up, and there's a hole, let the potassium through, and then they close again. So very simple structure, but a very complicated function. Now, there's another really intriguing way that ion channels will do the same sort of thing. Instead of stepping back to open and then coming together chest to chest to close again, let's just say that they all keep standing back and you've got this open hole. The ion channels, these four globs, can also have a fifth part to it, which is basically a long floppy strand of amino acids, so a long piece of spaghetti, at the end of which is got a gigantic ball. Okay, it literally looks like a ball and chain that you used to put on the feet of people, on criminals and slaves a couple hundred years ago. That ball and chain that's stuck on the ankle of one of those four globs, the big ball can kind of, as it's kind of moving around, can plunk into position in the middle of the hole, and that plugs the channel. So now the channel's closed. And then there's other ways in which that person can kick the ball out of the way, kick their ankle, and the ball falls out of the way. Now the channel's open. And then the ball plops back into place and it's closed. So it's a simple structure. It's just a ball and chain, and that becomes the open and closing. It's so simple, and yet it's so complicated.
Yeah. Wow.
Okay, if we had the time, we've talked about a lot of complicated things. I mean, we've talked about helices, pipes, and filaments, and beams, and sheets, and screws, and now we've talked about ion channels. I'd love to talk about, how these proteins, which again are just simply toothpaste coming out of a tube, that's what creates these parts that use those forces to adopt different shapes, and those shapes can then give you function. And so I'd like to talk about cutting tools and gears and propellers and walking machines, even little cells, nano tractors and trucks. You might think that those are really complicated, but actually once you begin to understand these principles that we've been talking about for 40 minutes now, it's really not hard to build those shapes. It is, on one level, I look at this and I think it's so simple and straightforward. And yet there is this immense amount of complexity and beauty to it. Yeah. So let's get back on track here. Next week, we're going to talk to Dr. Michael Behe about a whole series of protein parts that come together to make an engine, a rotary engine, which is simply just a series of mechanical parts. Whether you're talking about an engine in a car, or he'll refer to the engine on a boat, or the rotary engine at the back of the bacterium, all these scenarios, you're just pulling together bushings and pistons and shafts and screws, using the principles that we've just talked about. And again, those principles basically involve the sequence of the amino acids, which is determined by the genetic sequence, and how those amino acids interact with each other. That's all it is. Simple and yet so profoundly complicated. Right. So, I want to emphasize, for those people who think, okay, this really, it minimizes the Creator God because they see this just, I'm just dismissing a lot of really beautiful creative work, and I'm just saying, hey, it's just simple physics and chemistry, folks. I want you to think about a painter like Leonardo da Vinci. He just uses simple paints, he mixes them with a stick, a brush, and he brushes that on a simple canvas. There's nothing inherently complicated in what da Vinci does. It's just how those ingredients are combined to give you beautiful painting, and once again, open the door to design. Right. Oh. Well,
I mean, that's the issue in a sense of, what's the master builder? Like, what is the thing that determines that those shapes are going to be well, obviously it's the DNA and so on, but I guess what Behe is saying is, unless I have the idea of building a rotary engine, why would I build all these parts? Right. In other words, unless I have an end goal in mind. So what you're arguing, or what the more pure, let's say, Darwinian would argue, is that there is no teleology, there's no end goal. For whatever reason, these parts were created. They were maybe used in other areas, but suddenly, oh, guess what? They're useful for doing this too. And they kind of came together.
Right.
Like the mousetrap, right?
Yep.
But at the end of the day, what is the main point of saying what all that you had explained?
Right.
So go ahead.
And I guess in one sense, I don't have to say that I've made a perfect entryway for the intelligent design people, because you don't need a personified being.
Right.
You can still have inanimate forces that produce beautiful things.
Right.
So we've talked about the amino acids using inanimate forces to produce these beautiful things. Think about the geochemical and geophysical forces that made the Grand Canyon. Those are just geochemical and geophysical forces that created this great Grand Canyon. The physics that produces a beautiful sunset against, you've got this in the background, this stormy clouds and a rainbow in the sky. A beautiful scene, but it's just a bunch of physics, folks. It's just light physics and what have you. The physics that produces the beautiful swirls on a piece of granite or the colored crystals of a geode. Scott, do you think people know what a geode is?
I think so.
So you might have seen this in a tourist shop or a collectibles shop. Basically, it's a rock that's cut in half. And when you look at it from the one side, the uncut side, it just looks like a white blob, just an unattractive rock. But when you look at the other side, you see this beautiful design of colored crystals and things. It's just absolutely beautiful. And yet, when you understand the physics behind how those crystals form, it's really nothing complicated. So,
and I guess this kind of hits at the whole philosophical issue of reductionism, right? I mean, science is very reductionist, but I think once you reduce something to its simplest components, it does seem to take kind of the magic away from it.
Right.
But I don't think it needs to. Some people have called this nothing butterism. It's nothing but this, it's nothing but that, right? But that's not necessarily the case. I think, in fact, the fact that you can build such complexity and beauty out of such simple things is itself an amazing thing. I don't think that demeans it. I think it, in fact, quite the opposite. Even theologians have always argued, at least Christian theologians, that God himself is simple. That he's a simple being, which is kind of interesting. So the idea that nature is simple, that is made out of very few simple components, to me, that doesn't take away anything.
Right. Okay. That was a long explanation of how proteins are just like toothpaste coming out of a tube. And because of the sequence of amino acids, they generate these forces and these interactions and repulsions that form very specific shapes. And it's the shape of those proteins that gives the function.
Yeah.
So at this point, we could end the episode. There's a few other concepts that I do want people to understand, again, because they're relevant to when you hear ID people talking. It sounds more complicated than it needs to be. All right. So one of the things that they'll talk about is assembly. There's got to be, okay, now you just talk about how you made all these parts, but somebody has to put the parts together, right? And you think, okay, yeah, that makes sense. Somebody does have to turn the screws using a screwdriver, that kind of a thing. But when you talk about biochemistry, cell biology at the molecular level, a lot of those pieces know how to assemble themselves. There's this self-assembly. It's a common feature. It sounds like magic, folks, but it is a common feature in living cells. And so you don't need somebody, a designer, to turn the screws, tighten them up, and adjust the pieces. It happens by itself. It's like a crystal forming. So let me talk about how this happens. And it begins with a concept that I'll refer to as thermal energy. Okay? It's just another way of saying molecules jiggling. They're always vibrating. It's a very simple thing, but it's a fact. All these molecules are jiggling because they've got thermal energy. They're just nervous little things that are just quivering away. It's a physical property. And I don't think I need to say more, but let me ask you, Scott, do you think people are comfortable with this idea of all these molecules having this thermal energy, which just causes them to quiver and shake and vibrate?
Yeah, I don't, it's certainly not intuitive, I don't think. But I guess this is what we have discovered through scientific experimentation, right? That, in fact, that's the case.
Yeah. Okay, so that may not be quite familiar to most people. You may not have heard that idea, but it is a fact. You can look it up and Google it, learn what thermal energy is. It's basically all molecules vibrating. I'm just going to give you one example of where you will have already encountered this. Any smell in a room, whatever the molecules that are smelling, like a skunk, for example, or cinnamon, baking, the turkey's roasting in the oven, and you open up the kitchen door, the smell molecules are all jiggling. They're all vibrating, but it's more likely they're going to vibrate away from the central source than to keep going back and be reconnected to the original source. It's more likely they're going to move away. They're going to jiggle away from the source. And so that's why you put something like a cup of coffee in one end of the room. You will smell the cup of coffee. It might take a while, but that's simply because of the thermal energy of the coffee aroma molecules. And I'm going to have to trust that people can accept that. But this is important. Yeah. So let's now use this thermal energy to cause self-assembly. I want you to imagine, Scott, you're in the kitchen, you open up the cutlery drawer. This is the spoon cutlery drawer, and it's got a full set of spoons. They're all the same shape and size, but they're all loose and scattered in that cutlery drawer. And I want you to imagine that you start jiggling the drawer. All these spoons start jiggling, and that's the inherent thermal energy that they all have. They're all jiggling away. And if you can picture that, then I think you can imagine through chance alone that two spoons might come together. They just might happen to, the bowl of one goes against the bowl of the other. These kind of fit together, and for a brief period of time, those two will be together. But because you're jiggling, they're going to fall apart. Okay?
Right.
But they did stick together momentarily purely because of the odd shape of the spoon. The bowl against the other bowl and the handle against the other handle. They just kind of fit together. Briefly, they stuck together. It's a very brief thing, but it wouldn't be long. But now I want you to imagine in those spoons, you've built in little magnets. Okay? And the magnets have a north and a south pole again. And you put in the magnets in a way that the top side of the spoon is the north pole, and the bottom side, the outside of the bowl of the spoon, is the south pole. So now when the spoons start jiggling together, and it happens the two come together, bowl against bowl, the two magnets will quickly stick together. And if they came together backwards, the north pole against the north pole will push the two spoons apart. But when the spoons come together in the right way, the north pole, south pole come together, they stick together, and then you can imagine a third one sticking up against the second, and a fourth against the third. And so by simply this property of the magnetism in the spoons, you can start to build up groups of spoons in threes and fours and fives. It's just a matter of time, because you're jiggling the drawer and the magnets begin to align those spoons and hold the ones that fit together or push away the ones that don't. Right. Now, that's magnetism, but it's self-assembly using magnetism, but cells do that not using magnetism. They use those electrostatic forces. You can put a bunch of positively charged amino acids in one side of the bowl of the spoon or the protein, and negative charges on the other side of that protein, and simply they're going to stick together just like the magnets. And if you end up putting the proteins backwards, back to back, you push the negatives against the negatives, they're going to push those spoons apart. So in the same way, these proteins will begin to self-assemble, like spoons self-organizing into this stack of spoons, simply because you have thermal energy and an attractive or repulsive force. That's all there is. Nothing magic to it.
Okay. So let me see if I understand correctly. In the case of your analogy, you're making a simple analogy where spoons just have some magnets in them. What you've explained before is, in addition to magnets, there's many other little things in the spoons, let's say, that will determine how those spoons attach to each other.
Right.
And each of those spoons may have a different order of those things. But whatever that is, when you set that all up, if you take a drawer full of spoons and they're all set up with all those things, and you shake that thing up, every time it's going to assemble into whatever was determined by their inner structure. Is that pretty much what you're...
It is. And so, for example, when we do talk to Michael Behe about the flagellum, he might refer to the bushing, right? Even that is a technical term. So you can imagine a propeller on a shaft, and then around it, it's got to sit in a bushing would have the ball bearings to allow this shaft to spin smoothly around it, right? So he'll talk about the bushing, and he'll say the bushings are made of 8 proteins that spontaneously self-form to produce the bushing. Well, they self-form in the same way that those 8 spoons self-formed by their thermal energy, and then the right placement of positive or negative amino acids, or hydrophobic or hydrophilic amino acids.
Right.
So in the same way that a magnet has a north-south pole, the electrostatic has a positive and negative attraction, and the hydrophilic or hydrophobic attraction. And so by using these forces, the bushing self-assembles. Right. And then you can talk about how the shaft of the propeller self-assembles It's just simply a helix. If you can imagine, we were talking earlier about building up those amino acids into the shape of a helix. And if you have the right amino acid placement, that helix will want to be in the middle of the bushing that we just created. Right. And then a propeller can form. I talked about the 4 ion channels, the 4 globs that make up an ion channel, holding hands and opening and closing. Well, now imagine those globs are shaped a little bit differently, so that now they form the blades of the propeller, and that propeller just wants to be on the end of the shaft. It all begins to self-assemble and then self-function. It may sound like magic. It may sound like I'm making this up, but it can happen on its own.
It's interesting, maybe another analogy that comes to my mind, because I've mentioned a few times, I think, in our podcast, as a hobby, I build scale models. Let's say a model airplane. So when I get a model airplane, of course, the first thing I do is I open the box, I look at it, and there's a whole bunch of parts in there, right? A whole bunch of different pieces. And then guess what? There's an instruction book, right? And I take that instruction book, and that instruction book guides me through how I'm going to put all those pieces together to make this airplane. What you're kind of saying is the instruction book is built into the pieces.
In a sense.
And then there's in a sense, and there's a force outside called thermoenergy that causes those pieces to, like you said, self-assemble based on the instructions that are already built into them.
Right.
So there's no need for an external builder like myself to apply instructions, because the instructions are built right into the thing in the first place.
Right.
Okay.
So I want to say this is another analogy, but it's not really so much analogy. This is also the same basic principles, which is crystal formation, salt crystals. Yeah. And we talked about the geode, which are rock crystals. Those things form all by themselves simply because they have positive and negative charges, and there may be other factors that come into play here, but those crystals self-form simply because of their physical properties. And now we're talking about proteins that will self-assemble. It's the same principle. It may look different in some ways, but it's the same basic principles. They have these properties that they self-assemble. It does happen.
It's interesting because in the case of snow, for example, even though all snowflakes are in one sense unique, but nonetheless, a snowflake is always a snowflake. In other words, if you apply certain physical forces to water, it will always produce snowflakes. You don't need a divine intervention to produce them. They will be produced, and they will be produced as snowflakes. So I guess, I can understand the analogy.
Okay, so Scott, let's move on. We've spent a long time talking about amino acid sequences giving you structures, and that's based on the genetic sequence, of course, and how those structures can then give you all kinds of shapes, which then give you function. Yeah. And then the past 5 or 10 minutes talking about self-assembly. It does happen. I want to talk about a third basic mechanism, again, that you kind of need to know when you listen to the ID people talking, which is information in the genetic sequence. Now, Darwinism basically talks about how you have individual mutations, and each change now is a new form of information, and whether that's good or bad. But the main point I'm getting at here is they will talk about individual changes, point mutations. You've got a long stretch of DNA with 1,000 bases, and base number 682 now changes to something else. That one out of the 1,000. But there's a lot more to evolution than just that. There's massive change in the DNA where you're not just changing one base, but you're moving around large chunks of DNA. This happens all the time, and we talked at length about that in episode number 70 with Dr. James Shapiro. So I'm going to just ask the listener, if you want to know more about that, go to episode number 70. But enough to say that you don't only have to think about, or you shouldn't only think about one individual point change at a time. You can have a massive movement, kind of like moving chapter 13 and putting it in front of chapter 4, and then taking out, the third page of chapter 7 and moving it to the end of the book. That kind of rearrangement, not just simply changing individual letters. Right. And the other thing that I want to emphasize here is that in addition to moving large chunks of DNA, what often can happen is that you get duplications of these large chunks. So duplication of a gene, or duplication even of a whole chromosome. And so then you move that duplicated copy somewhere else, and that duplicated copy can now basically serve as an R&D factory, a research and development factory, where you can now play around with those gene sequences, the duplicated ones. You don't have to worry about the first set that you need for example, to code for the proteins of the flagellum, which is what Michael Behe will talk about, you have this duplicated set of genes that you can now play around with and turn those parts that became a flagellum, turn them into enzymes, turn them into ion channels. Of course, I'm really, that would take many steps, but you can make massive changes in the genes simply by creating a copy, a duplicate copy, and start messing around with that. So when they talk about these cataclysmic changes, as soon as you make one change in a sequence, now you've disrupted the whole protein and you've lost function. Well, you have not lost function in the original copy that you hang on to. In the duplicated copy, you've lost the function as the mousetrap. They'll talk about the spring. It's no longer a spring. You're right, but now maybe it's a wire and it can serve an electrical circuit, that kind of an idea. And we refer to that as co-optation, that protein can now be used for a different purpose. And sometimes that can be the exact same protein can be used for multiple purposes. Or you can take a protein, make a minor change in it, and it no longer really works too well in that purpose, but now it's a great something else. So you really need to understand that whole concept.
Yeah.
So Scott, we're moving fast. I've got so much material and I can't use it all. We've talked about the amino acid sequences. We've talked about self-assembly. We've talked about moving large chunks of DNA around. Now I want to talk about modular building up of proteins, building up molecular machines and tools by swapping different parts around. I want to start with a simple combination of two simple coding sequences. Okay, we talked about building that long pipe, the helix, and we talked about building a sheet, the beta-pleated sheet. We talked about that earlier. I want you to imagine taking that sheet and putting it at the end of the helix. So in other words, you put together the piece of DNA that coded for the helix and the piece of DNA that coded for the sheet, string them together end to end, and now when you pump out that protein, it's going to spontaneously form into a long pole at the end of which is a sheet. And now you've basically got yourself a spoon or a shovel, right? A long handle with a blade on the end. And if you want, we'll attach another little loop at the first end, at the front end of that, to be the handle. So I'm putting together three distinct segments of DNA, one that codes for the loop on the handle, one for the handle itself, and one for the blade. Pump out those proteins. They're going to spontaneously form to give you spoons or shovels, all right? And then you can tweak the DNA a little bit to now make the blade a little bit curved, like a spoon might be or like a shovel might be. Those are just minor tweakings. But now I want you to imagine that you take the section that coded for the bowl of the spoon or the shovel and replace it for another one that codes for a different flat piece, one that's more extended and has a sharp edge to it, a sharp ending. Now instead of a spoon or a shovel, you've got a knife or an axe. You can make another change, and instead of the spoon or the knife, you replace that third piece of DNA for something that gives you a pointed ending, a very sharp spike, so that now you've got a needle or a spike, a stiletto or a poker. Maybe you add several versions of that together and do it in a way that now at the end of the handle, you don't have one spike, you've got three or four. Well, now you've got a fork, either for eating or for pitching bales of hay. Or instead of spikes, you just have a big globular, a stretch of DNA that gives you a globular end, a big chunk at the end of the handle. And now what you've got is a hammer. So by simply taking these long stretches of DNA, you can build up a spoon into a knife, into a fork, into a hammer. It's just simply by moving pieces around and then tweaking the genes in that. Any thoughts, Scott?
Yeah, it's amazing, that from such, well, I mean, it's amazing in one sense, but in another sense, we do it all the time in the real world. We build up things from small parts and reuse things and so on. So it is intuitive.
So the cell will actually do this exact sort of thing. It happens in your body all the time, and your immune system takes it to a whole new level. You've got genes for the antibodies that make our immune system so powerful and so complex. It's got regulatory mechanisms in place where it's just moving large pieces of DNA all over the place to form entirely new molecules. Now, it'll keep more or less in the same condition the stretches of DNA that give you the main body of an antibody, which, if you want to think about it, folks, an antibody is just basically a long arm at the end of which is a hand. And at the fingertips of the hand will be very unique sequences of amino acids, shapes of protein that will identify this is a viral toxin versus this is one of your own blood cells. So your antibody is this long arm with a hand and then the really sensitive fingertips. And the immune system will keep the coding for the long arm more or less intact because it needs that long arm. That's how it grabs onto things and pulls them out of the body, essentially. But it shuffles around the DNA that codes for the hand, and more importantly, the DNA that codes for the fingertips. It's just shuffling the cards like it's on steroids. It's just shuffling them all over the place. Comes up with millions of combinations of little proteins at the fingertips, and then it just starts throwing them at the virus. And out of the millions of different shapes, 1 or 5 or 10 will be just the perfect fit for that viral protein. And then there's a mechanism in place that says, okay, you got it, you're the one, and they start making multiple copies of that set of DNA and start pumping out antibodies, and that's how you kill the virus. So it's really this idea of shuffling around chunks of DNA to give you entirely new shapes. The immune system that takes that into I want to say cosmic level is just shuffling DNA like crazy to produce all these different antibodies that give you immunity for your whole life.
It's like it's almost using evolution itself.
Yeah.
Like in a sense, right? Like it's just like you said, shuffling the DNA to come up with, in one sense, it's a brute force method, isn't it? Like it basically, I'm just going to create every combination possible kind of thing, and I'm going to throw it at the wall and see what sticks.
Exactly.
Right? And yeah,
exactly. So, Scott, we've really, really gone on long. We've covered a lot of ground. I hope that this will be useful when the listeners listen next week to Michael Behe, and at some point we'll bring in John McClatchy as well. Basically, we've talked about how the gene coding, the gene sequence, gives you the amino acid sequence. The amino acid sequence produces these internal forces that cause the long stretch of amino acids to take on a certain shape, and that's the protein. And those shapes can self-assemble and produce bigger shapes and ever more complicated shapes. And then those complicated shapes can self-assemble. And that's how you get all these really beautiful and wonderful machines inside the cell. And a lot of it's just driven by, I see it as all driven by inanimate forces. There's not a little tinkerer sitting there with a screwdriver and a hammer in hand putting them together. Those pieces self-assemble. And we've talked also about, modular creation of these different proteins. We've covered a lot of ground. I've basically given you a Biology 101 lecture in about an hour and a half. But I think you do need to have this material at hand when you listen to our ID advocates.
Well, I've thoroughly enjoyed it, and it's certainly helped me understand at a certain level, obviously at a very high level, what people talk about all the time, our genes and how we're put together. I'm empathetic, or I'm sympathetic to not just the ID movement, but people who invoke teleology in science, because it is amazing, and certainly at first glance, you look at the complexity of the cell and it looks designed. How could you come up with that any other? And yet, as you've explained, it's amazing what nature is able to accomplish through natural forces.
Right.
At a very basic level. So yeah, very good. Really enjoyed it.
Next week, we'll talk to Michael Behe, and in a couple of weeks, Jonathan McClatchy about intelligent design.
Excellent.
Thanks, Scott.
Thank you.
I've kept you too long. Have a good day.
Yeah, you too. Take care.
Okay, ciao.
Bye-bye.
You've been listening to Recovering Evangelicals with your hosts, Luke Jansen and Scott Dyer. Send your comments and suggestions for new episodes to lukejjansen@gmail.com. Join our private discussion group on Facebook by searching for the Recovering Evangelicals podcast group. Look for the logo that you always see at your favorite podcast catcher and ask to be let in. Subscribe to lukejjansen.wordpress.com if you want to get advanced releases and group posts. Or browse through our archive for a full listing of all our previous episodes. And please help the podcast by sharing with a friend and leaving a review at your favorite podcast catcher.